So now that we've talked about how we target proteins to the ER, let's take this a step further and explain how we can manipulate this process to actually insert proteins into the ER membrane. And I want you to remember here that any protein that is in any membrane originated through this process. So it came from the ER originally. So again, we have our signal sequence, which is a series of hydrophobic amino acids, and this is a key part to it. There are basic residues that might be on the front or on the back, and there may or may not be a signal peptidase. And this entire thing may be at the beginning of a protein, it may be in the middle of a protein. And we can end up with proteins inserted into the membrane in a number of different ways. For example, we could have a protein that just has a single alpha helix spanning the membrane, and we have the N terminus on the ER lumen side and the C terminus on the cytoplasm side. Or we could have the exact opposite. We could have a single spanning alpha helix with the N terminal on the cytoplasmic side and the C terminal on the ER lumen. Or we could have something much more complex, a multi-spanning process that has several helices going through the membrane. And again, we might have the N terminus on the lumen side and the C terminus on the cytoplasmic side. We may see both termini on one side or the other, or we could have this whole thing flipped. So there's lots of different possibilities here. And this is where the book kind of stops, but we can take it a step further and actually show what the classification system is for these proteins. So the very first thing when we talk about targeting the protein to the ER, we brought up the idea that we have a signal sequence that is cleaved off. If we have that type of paradigm, we would call that a type 1. But type 2 and 3 lack the cleavable signal sequence. And in this case, it might be that transmembrane region, which would be hydrophobic, that is the signal sequence itself. And here we can see we have type 2, which has the N terminus on the cytoplasmic side, where type 3 have the N terminus on the endoplasmic reticular lumen side, so on the inside. We could also have the scenario where the signal sequence is actually at the very end of a protein, and in this case, it is handled through a slightly different process, and we'll talk about that in more detail here. Here we have type 4 proteins, which have multi-spanning, and this covers all multi-spanning, and it can be broken up into subgroups, of course. And there is one last style of proteins anchored to membranes, and that's through something called a GPI anchor. And we'll talk about that here towards the end. So, looking at these from a sequence standpoint, here we have all these different types lined up, and we have the N terminus marked and the C terminus marked. And here we show type 1 that has a signal sequence, and then we have some type of internal sequence as well, which is where the transmembrane portion is going to be. Here, the positive amino acids are actually present in the signal sequence. On type 2 and type 3, we can see that we don't have a separate signal sequence. We just have this internal, what we call a signal anchor sequence, because this is both a signal and an anchor point. Over here, we have a separate signal, and then we have a second separate anchor sequence. Here for type 2, we can see that the positive charges are on the cytoplasmic side. And here, where it's flipped out or flipped upside down, you can see that the positive charges are still on the cytoplasmic side. We're just marking up here which side is in the lumen, which side is in the cytoplasm. These positive charges are actually directly responsible for the orientation of the protein in the membrane. Here we have a couple examples of multi-spanning sequences. And here you can see that not all of the sequence have these positive charges around it. So you can see this one here does not have a positive charge. But the orientation of this first one is going to directly dictate the orientation of the next one and the next one and the next one. So we don't necessarily need all those positive charges. They may or may not be present. Here we have one that has 4 spanning domains, and what this means is that both the N-terminus and the C-terminus are going to be on the same side. Here you can see that it's both on the cytoplasmic side. If you move these charges over to this side, you would have both termini present in the lumen side. Here we have 1, 2, 3, 4, 5, 6, 7. So it's an odd number of transmembrane domains. And here we can see, because we have the charges after the signal anchor sequence, the N-terminus is going to be in the lumen, but since we have an odd number of transmembrane domains, the C-terminus is going to be on the cytoplasm. Again, it would be very easy just simply to take these positive residues, move them over to this side, and then we would have the N-terminus be in the cytoplasm, and consequently the C-terminus would be in the lumen. So these positive charges make a very clear impact. The thing that these signal anchor sequences or signal stop transfer anchor sequences have in common is that they are all hydrophobic. And what they form is an alpha helix that will span across the membrane. And if you remember, in alpha helix, all of the amino acids, all of the R chains are pointed outwards. So if you have a cylinder that the entire outside of it is hydrophobic in nature, you could see how that could easily span a membrane. And we can actually utilize this by taking an algorithm and scanning protein sequences and look for what might be recognized as a signal sequence. So something that has a particular number of amino acids and has positive charges to one side or the other. And then later on down the sequence, we have another region which is hydrophobic. So what this scale is showing is the higher the number is, the more hydrophobic that region is. The lower the number, or the more negative the number, the more charged or hydrophilic that region is. So here we have a protein that is got a lot of hydrophilic region and a couple of very key hydrophobic long regions, right? So this is long enough to fit across a membrane. This right here is long enough to serve as a signal sequence. So if we were to look at the primary structure of a protein that we can deduce from the DNA sequence, we can make certain predictions on, okay, I think this might be a transmembrane protein. This gets even easier when we have multiple transmembrane domains. If you look at something like this, there's little doubt that this is a spanning protein. So now that we have talked about that signal sequence, let's talk about how it actually works. So again, here is the ribosome, here's the messenger RNA. It starts producing this protein. On this protein, it just so happens that the very first thing is the signal sequence. You produce that charged residue. That charged residue actually associates with the side of the ribosome. And then we have that long hydrophobic region. And that hydrophobic region is going to be recognized by something called the SRP, or signal recognition protein. And this protein will act as a bridge, binding what we call the SRP receptor. And this SRP receptor is associated with the translocon pore. You can see here at this point, the translocon pore is closed. Also associated with this pore, or this translocon, is the signal peptidase. This was what will cleave off the signal sequence if it does have a protease site present in its sequence. So this will associate all together. The pore will then open up, and we will see this growing polypeptide chain being exported or moved into the pore. And within this pore, we actually have a subregion which binds that hydrophobic span. Okay. In this case, we have that signal sequence at the front. It is going to be cleavable, and so what we do is we continue secreting this protein. It starts to loop into the ER, and it will start folding on the inside, but it's going to recognize the cut site, and the protease will cut the protein there. The protein, once it's finished being secreted, will fold in the ER itself, and this little leader sequence or signal sequence that is cleaved off will ultimately be moved out and then recycled. So now let's take a little bit closer look at the actual structure of this translocon protein. It is actually made up of 3 separate subunits. So we have the gamma subunit, the alpha subunit, and the beta subunit, also known by the acronyms or sec-Y and sec-E. And all of these have transmembrane spanning regions. So this is looking down at it from the top, kind of like if we were the ribosome about to dock with this ring. And this is actually where the pore is. And here we can see that this entranceway is blocked, that the pore is not open, it is closed. Here's looking at it from the side on how it would appear actually fit into a membrane. And you can see that we have transmembrane regions here. We have this ring in the center, and we can see here that it's closed. So the thing that determines whether this is open or closed is this short alpha helix attached to this loop here. When it is in the closed position, that alpha helix is up, blocking the pore. When it is open, ready to allow passage through, it is moved over here in this position. So it moves from here over to here. And you can see that this has kind of an hourglass type of shape, and the hole or the pore in here is very small and essentially just large enough to allow an unfolded protein to pass through. Okay, so now we have a little bit closer view on what that looks like, what that translocon protein looks like. Next, what we'll do is take a look at the signal recognition particle and its receptor. So we already know that the SRP has a binding pocket for this hydrophobic region in the signal sequence. We also know that it has a binding site for the SRP receptor, because these two form an association. So let's go into a little bit more detail about that. So here is an image of the SRP, and what we see here is a very large hydrophobic groove. And it's this groove that interacts with the hydrophobic residues on the target protein, or whatever protein is being produced that has this hydrophobic region. Here we have the SRP, which is shown on top, and on the bottom we have the SRP receptor. And you can see how they are bound together. One thing that you might notice right away is that both of these are also binding GTP. So the SRP molecule has a GTP binding site, and the receptor itself has a GTP binding site. And what's important about this is that these two will only interact if they are both bound to GTP. Okay, so GTP is a requirement in this association. The added relationship that we have here is when this hydrophobic groove is bound to a target receptor sequence, the GTP binding site is open. When it's not bound to it, it cannot bind GTP. Consequently, the SRP cannot bind the receptor, right? So as soon as it interacts with a hydrophobic region, GTP binding site opens up, GTP binds, and then we have the ability of forming this conjunction. So here's an image of that. So we have the ribosome here on its messenger RNA. It starts producing a protein. This one happens to have a signal sequence at the N-terminus. It is produced. It associates with the SRP. That allows SRP to bind GTP, and the SRP receptor is also bound to GTP, which allows them to form a unit. Now, what happens is this brings the ribosome in close conjunction with the translocon, and consequently, these GTPs are hydrolyzed and are released. That causes the SRP to fall off. It also disassociates the SRP with the region, and this conjunction also opens the translocon. So here in one movement, we get the hydrolysis of GTP, we get the ribosome sitting down, the pore opening up, and the signal sequence being released, allowing it to go into that pore. That starts to move through the pore, and if there is a protease site, which this protein shown has, a protease would then trim off the cleavage point. What is not showing here is this leader sequence is typically up in that hydrophobic pocket of the SRP that we talked about before. Once that is removed, we then get continued secretion. Now, coming back to the idea of how things progress through this secretion pathway, like in a traditional sense. So, we have things that are produced in the endoplasmic reticulum, and it gets transported in vesicles to the Golgi apparatus and ultimately to the outside. Something that I want to point out now that we've thought about the fact that proteins get inserted into the membrane, they're inserted in a very specific orientation. I want you to recognize that here we have a protein that's inserted into the membrane. It has a big globular domain on one side and a small side on the other. So the inside of the lumen the lumen of the ER is continuous with the inside of the vesicle, which is the same space as the inside of the Golgi apparatus and all of its various stacks, and ultimately is the same as the outside of the plasma membrane. So whatever is on the inside will become the outside, or will remain on the inside if it's in any type of vesicle. You can imagine if this was then transported back to, let's say, the nucleus, and it were to fuse with the nucleus, the small side would be on that inner membrane space in the nucleus. So just kind of keep a mental map of the orientation things are going through, and that if it's facing the inside of the endoplasmic reticulum and it's moving to the outside, whatever is facing the inside will be the outside of the cell. So we've talked about how the association is formed, but now let's talk about how we can actually insert the protein into the membrane. So here we have an image of a protein being produced, and this protein is in that type 1 category that has a signal sequence and then has another transmembrane sequence in the middle of it. So it's starting to be produced, it has a signal sequence, it associates with the SRP, it binds the receptor, it goes through its GTP hydrolysis and all that. And now we have the signal sequence in this pocket, and it is cleaved by this peptidase. At this point, the signal sequence is actually released into the membrane itself. There is essentially a side door in this channel that allows a hydrophobic structure to be released into this hydrophobic space. So in this circumstance, that leader sequence is released, and ultimately it will be degraded by proteases that are associated with the membrane, and that will be recycled with its amino acids. We now have the rest of the protein being produced. It comes to the second stretch of hydrophobic residues. Those hydrophobic residues essentially bind into that same hydrophobic cleft that is in the translocon. And this time, since there is no cleavage site here, the peptidase doesn't do anything, and it's just simply released through the side door. At this point, the rest of the protein will continue to be produced. We'll get dissociation of the ribosome from the translocon, and ultimately, when it's finished producing that protein, it will dissociate, and now we have a freshly inserted protein into the membrane. So this is how we deal with class 1 proteins, right? So these have a leader sequence that is cleavable, and then there's another hydrophobic transmembrane sequence in the middle. Now let's go on and take a look at some of the other types. So here we have a example of what's happening with a type 2 protein. In a type 2 protein, we have or a type 2 transmembrane protein, we have the N terminus is going to end up being on the exterior, and the C terminus will be on the interior. So in this circumstance, what happens is there is an association of that positive charge with both the water that's out here and also this ribosome itself, and that's going to contribute to which direction the SRP is binding, because the SRP is only recognizing the hydrophobic sequence. So it could bind onto that sequence in 2 different directions. So having that positive charge towards the N terminus will link that N terminus to the exterior, the SRP will bind to it, and then what you'll see is a loop coming down through. The N-terminus will retain outside. You'll get the binding of this internal sequence, and it will get shunted out. We'll continue to get the rest of the protein excreted through this pore until it's finished, and now we have the C-terminus inside. This is the exact opposite of type 3 here, where we have the N-terminus on the inside. And here what happens is we are producing this protein already. It has the positive charges on the other side that directs the SRP to bind in the opposite orientation, and that will essentially force the N-terminal loop to be pushed down through. And that will allow this association of that transmembrane sequence with that hydrophobic pocket. It'll take its side door, and then this is very similar to the type 1 circumstance. But here, the SRP is really directing the directionality of it, that the SRP in conjunction with those positive residues on the protein itself. Here we have an example of a multi-spanning protein. So this has several alpha helices. And here we get the exact same association happening, whether it's the N-terminal sticking down through or the N-terminal retained on the outside is all about which side the positive charges on are in this hydrophobic sequence that will direct the directionality of the binding and direct which side goes through. Here, the N-terminus is going to be retained on the outside. So that first part goes through. When it gets to the second transmembrane sequence, it will naturally bind that hydrophobic pocket. It will then get shunted out. Here we have a third sequence coming through. That third sequence will bind that pocket, get shunted out, and you can see we're forming loops with the rest of it. And here you can imagine this might continue on for a while, or let's say it ends here. And if it ends here, then this C-terminus will just come through and be remained over here. If there was another sequence, and we had an even number of transmembrane sequences, then we'd have both the N-terminus and C-terminus on the same side. All right, here is a slightly different scenario where the transmembrane sequence is down on the C-terminus of the protein. So here, by the time we actually produce a sequence that the SRP would recognize, the protein has already been produced. That's going to require a separate system to import it. And so here we have this complex here called TRC40. That will bind this hydrophobic region. That will associate with a different pore complex called GET1, GET2. That is going to require a little bit more energy to pull this whole structure through. So it requires a little bit of a different process. And then what we see here is a protein that essentially has its entire C-terminus buried in the membrane. All right, the final thing that I wanted to cover in this section is the concept of GPI anchored proteins. So in this process, we take a protein that has already been inserted into the membrane. And what we do is we essentially cut the protein off and couple it onto what we call a GPI anchor. And what this GPI anchor is essentially a couple of fatty acid chains cross-linked to a series of sugars. And this is just acting as essentially a way of cross-linking a protein onto a phospholipid. The reason why the cell does this is there, when you have something that spans the entire membrane, it does place certain constraints on where that protein can show up. So the membrane can actually be thicker in some spots and thinner in other spots. But the length of this alpha helix that is passing through will limit this protein to be in the region that matches the length of this hydrophobic alpha helix. So by detaching it from that and instead attaching it to this GPI anchor, what we can do is still keep the protein bound to the membrane. But this is now coupled to a phospholipid, and that phospholipid can be varied. This means that we can make several different versions of this protein that might be able to fit into several different regions of the membrane itself. So you could take a GPI anchor with slightly longer and it would fit into one region. And this gives just a way of being able to moderate where and when this particular protein shows up. Now, the fact that this modification occurs within the lumen, so we're attaching it onto the leaflet of the membrane that is facing the lumen. This means that if this protein is exported out to the surface, it would actually be facing outwards. Okay? This is the only other way of having a membrane-bound protein that is outward facing on a cell. We've talked before about modifications where we just simply attach a fatty acid onto a protein in order to associate it with the membrane without actually being a transmembrane protein. And that is something that is completely limited to the cytosol. So if we want things to be peripheral membrane proteins, and we want it to be on the cytoplasm, we can just add on a fatty acid, like palmitylation, which we discussed in protein modification. But if we want that to be on the exterior leaflet of the cell, that would have to be accomplished through this GPI anchor. So now we have a good idea of how proteins are inserted into the membrane.